start here by looking at the blood supply to the kidneys. And so the blood supply to the kidneys is coming in from the renal arteries, coming off of the aorta. So the abdominal aorta gives rise to the renal artery. The renal artery will then branch into both kidneys on either side. And then it starts to divide as it moves through the medulla and then eventually to the cortex. So the first branch from the renal artery is the segmental artery. And the segmental arteries are going to each sort of general segment of the kidney. We have 2 or 3 main segmental arteries here. That will then branch into the interlobar arteries. And the interlobar arteries are going to be going in between each of our renal pyramids, running in the renal column. So remember, the renal column is the space in between our pyramids, and this is where we find the interlobar arteries. That will then give rise to the arcuate arteries. And just like the name implies, the arcuate arteries are going to curve over the top of the base of the renal pyramid. So the broadest part of the renal pyramid here is where we have the arcuate arteries. That then branches into the cortical radiate arteries, which are these very tiny distal branches that are communicating toward the nephron, all right? Remember, the glomerulus and the proximal part of the tubule, the Bowman's capsule, those structures are found out here in the cortex. And so this is where the radiate arteries, the cortical radiate arteries, are going to be projecting towards the glomerular structures. That then communicates to the afferent arteriole. So that artery, or that very small branch of the cortical radiate artery that we drew, that afferent arteriole going toward the glomerulus, is coming off of the cortical radiate artery. So it's not being shown here, but the way that we drew it out is then communicating immediately to the glomerulus. That then gives rise to the glomerular capillaries, that ball of capillaries that's stuffed down inside our glomerular capsule. And this is where we begin to filter the waste products that are being dropped off from the blood, and those will then be filtered through the nephron to begin forming our filtrate. Let's look at some of the venous structures here. So after that waste is dropped off in the nephron, we then have these vessels branching toward the venous structures and sending that blood back towards the inferior vena cava. So we start with the efferent arteriole, so that branch, that vessel that's moving away from the glomerulus and back towards the venous structures here. Then we go to the peritubular capillaries, or the vasa recta. And so the peritubular capillaries are coming into contact with different aspects of the nephron. The peritubular capillaries is going to be closely associated with the proximal convoluted tubule and the distal convoluted tubule. They're actually going to wrap around these tubular structures, whereas the vasa recta is going to come into contact with the loop of Henle, okay? So these very small capillaries that are coming from the efferent arteriole are then going to then closely associate with these tubular structures. And this is how we reabsorb substances. This is how we take substances from the tubules, right, from the peritubular space, from the proximal convoluted tubule, the distal convoluted tubule, from the loop of Henle, and we can reabsorb that back into the blood, back into these capillary structures, and we can prevent those substances from being lost in the urine and send them back into the vascular circulation. That will then send blood to the interlobular veins, right? The interlobular veins, which are the veins that are very distal out here. So these are synonymous with the cortical radiate arteries. So there are no interlobular arteries. Instead, there are cortical radiate arteries, which are synonymous with the interlobular veins, the very tiniest vessels in the cortex here. That will then communicate to the arcuate veins, so the veins that wrap around the base of the renal pyramids. Again, this is a synonymous structure with the arcuate arteries. And then that communicates to the interlobar vein, the vein that is running in between the renal columns here. That then communicates to the renal vein, and then that goes right back to the inferior vena cava, okay? So that is how blood goes into the kidneys, is filtered through the glomerulus, and then makes its way back out into the inferior vena cava. Now I want to point out again, looking at our nephron and then adding that association of those vascular structures. So we talked about our proximal convoluted tubule. We talked about our renal corpuscle, which is made of the Bowman's capsule and the glomerulus. So we can see the glomerulus kind of being shoved down inside of that corpuscle here. And these are all structures that are located up in the cortex here. Now we want to understand how those vessels come into contact with these tubular structures. So the peritubular capillaries are wrapping around the proximal tubule and the distal tubule, whereas the vasa recta is wrapping around the loop of Henle, okay? And we can see how the loops of Henle dip way down into the medulla here, and then curve all the way back up and communicate to the distal tubular structures, and then to the collecting duct, which then dips back down into the medulla and towards the minor calyx. And we're looking at a histo micrograph here, so kind of the histology of the nephron, and we can appreciate some of those structures here. So here's what the glomerulus looks like from a histological view. Here's what some of the tubular structures look like. We can see the proximal convoluted tubule here. And what we're seeing is a cut surface of this tubule. So we can see the cells that are lining this tubule. And then we can compare that to the distal convoluted tubule. And we know that this is proximal versus distal because of the shape of these cells, right? The proximal tubule is really important for reabsorption. So it's going to have a lot of long or elongated cells, whereas the distal tubule has very short or flattened square shape or cuboidal cells. Getting into some of the other structures here, so some of the distal structures that we can see on gross inspection, we talked about the collecting ducts that are making their way from the distal convoluted tubule and communicating toward the minor calyx. So we've got the minor calyx here, which are the beginning of the duct-like structures here. That leads to the major calyx, or the larger branches of these ducts. And then that leads to the renal pelvis, which is the opening of the ureter, which then goes down to the urinary bladder, and then eventually to the urethra. So let's add some layer here. Let's add some more detail, and we're going to talk about some of the functional aspects to each part of the nephron. And we're going to continue doing this. This will be a revisiting theme. We're going to keep coming back to each part of the nephron and speak about the function that each part is responsible for in terms of the overall fine-tuning of that filtrate to become what we know as urine. So the proximal convoluted tubule is responsible for reabsorbing. It's going to be unregulated reabsorption. So it's going to reabsorb pretty much everything. It's going to reabsorb important nutrients, such as glucose and amino acids. It's going to absorb ions, such as sodium and chloride and potassium. It's going to reabsorb any type of plasma protein. So protein should not be in the filtrate. We'll talk about why that is going forward. But in the event that proteins do end up in the filtrate, the proximal convoluted tubule will reabsorb those things back into the vasculature. It's also going to absorb a lot of water. So because we don't want to lose most of the fluid or the watery component of our plasma, we want to preserve that as a part of our hydration, regulation of our hydration. The proximal convoluted tubule is going to reabsorb most of that water back into the bloodstream. All right. And so it's going to do all of that because of its association with the peritubular capillaries. And so I talked about this connection, this interaction between the proximal convoluted tubule and the peritubular capillaries that are wrapping around that tubule. And so this is how we can reabsorb important substances that made their way into the filtrate and reclaim those substances or reabsorb those substances back into the blood because of this connection with the peritubular capillaries wrapping around the proximal and distal convoluted tubules. Looking at the loop of Henle, so we'll start by looking at the descending portion. So there are two limbs to the loop of Henle. There's the descending limb and then the ascending limb. This is where we mainly absorb water. So most of the water that is reabsorbed in the kidneys is done through the loop of Henle, but this is through an entirely different mechanism that we will speak about going forward. And so the descending part will reabsorb water. Again, because of that interaction between the vasa recta, which are the capillary structures that are winding around the loop of Henle, and so the descending limb reabsorbs water, whereas the ascending limb is going to actively pump or actively remove sodium ions and chloride ions from the loop of Henle, from the tubule into the vasa recta, in order to prevent the loss of those important ions into the urine, okay? So the ascending limb is going to actively pump sodium and chloride out into the vasa recta, into the vasculature, whereas the descending limb is going to remove water, right? It's going to reabsorb water. Now, the most distal structures are going to be the distal convoluted tubule. So the other winding part of the tubular structures here, that is going to be involved in regulated reabsorption. So this is going to be very tightly regulated. It's going to be very selective to what is being reabsorbed and how much of it is being reabsorbed, unlike what we saw in the proximal convoluted tubule. So we're going to see selective reabsorption of sodium and calcium. We're also going to see secretion of ions, such as bicarbonate ions, as well as acid, right? Any type of acid, usually hydrogen ions, but any type of acid, as a way to balance the acidity of our blood, we're going to actively secrete ions and acid from the distal convoluted tubule, and very little water reabsorption takes place, okay? So most of the water reabsorption happens in the loop of Henle, as well as the proximal convoluted tubule. And then lastly here, we have the collecting duct. So the collecting duct is basically kind of fine-tuning that filtrate. It's going to just drain the rest of that filtrate, from the distal tubule toward the minor calyces and eventually major calyces. And it's not going to make a lot of adjustment, but it's going to make any final adjustments really having to do with water. So there are specific cells located here, the intercalated cells and the other cells we'll talk about. Those cells, the principal cells, those cells are going to fine tune this filtrate and reabsorb anything else that may be needed, or secrete anything else that may be present in the blood that we want to get rid of. And so very small fine-tuned adjustments are made in the distal, in the collecting duct, and then that filtrate can be sent to the minor calyces and then eventually to the major calyces. Okay, here is a very good overview of all of that. So this would be an excellent study tool in terms of summarizing some of the structural components of different aspects of the nephron and the collecting system, as well as some of the functional aspects. So I would encourage you to kind of recreate this, or maybe just print this out and add additional details as we go along. But just to review this, we've got the proximal convoluted tubule. This has long columnar cells, which have microvilli. And so this part of the tubule is really adapted for reabsorption of organic nutrients, amino acids, glucose, water, ions, so a lot of unregulated reabsorption. We've also got the distal tubule. This is involved in secretion. So anything that's present in the blood that we still want to get rid of at this time, such as toxins, byproducts or medications we're taking, drugs, acid, we can sort of secrete that from the blood into the distal tubule in order to get rid of those harmful or toxic substances. We then have the Nephron Loop, so the loop of Henle, which is that area that is dipping down into the medulla. This is where we mainly reabsorb water. So the descending part reabsorbs water. The ascending part is going to pump chloride and sodium out of the tubule and into the blood. And then we've got the tubular structures, which are the collecting ducts, and the end of that leading to the minor calyx. Okay, and then the beginning part, which is the glomerulus, which we talked about as well. Okay, so we're